Hello, good day. Thanks for coming to the talk today. My name is Trevor Brown, and I'm the Vice President and General Manager of LUMICKS Americas. I'm going to be your host for today. We have a very big week in store here at BPS and for the community at large as we officially announce a new tool that has been released and several years in the making. This tool is sure to accelerate discoveries and insights into how biological systems work at the smallest scale. I'm not going to ruin the surprise just yet, however. Today we also have two additional speakers. Dr. Trey Simpson, who is head of our amazing America's Application Scientist team, who's going to run through all the cool new features and how they address the real challenges in the field today. We'll also hear from a guest speaker, Dr. Arne Gennerich from the Albert Einstein College of Medicine, who will be talking today about his work on microtubules and motor proteins. First off, let me explain and tell you who we are and what drives us as LUMICKS. We are an innovative life science company building some of the most important tools to observe and understand the mechanisms and forces within biological systems at the smallest scale. We're a company of scientists, of physicists, and of engineers who want to revolutionize how the study of biological systems are done. So within the field of single molecule research, there are many great tools that the community has to help understand things like structure, such as cryo-EM and NMR, tools that help us understand function, bulk and activity-based assays. However, there's surprisingly little that reveal the mechanism of action and how single molecules behave in real time. Tools that allow us to dynamically interact with and observe the systems being studied. In order to get the full picture, you need to understand how it all works in context with correlated data sets. Together, we've been on this mission to democratize dynamic single molecule tools to ensure that researchers can access the unique and powerful capabilities of our system so they can focus on their science. Building off of years of knowledge from our founders and collaborators who work with home-built systems, we have developed a powerful and easy-to-use system, our flagship product, the C-Trap. The C-Trap is a convergence of optical tweezer, microfluidics, and powerful imaging technologies, seamlessly integrated and controlled by a unified software tool. In addition to offering an incredibly easy-to-use platform, we're supporting an entire ecosystem by developing a wide array of applications, biochemical reagents, and powerful data analysis and scripting tools that will save our researchers countless hours. What's more, our customers are able to connect with a rapidly growing user community to share and learn from one another. All you have to do is focus on creating impactful science. And impactful science is what happens when our customers use the C-Trap. Our customers published over 39 highly impactful articles in 2021. Highly impactful science. Let's take a look at just a few of the applications enabled by the C-Trap. Understanding DNA replication repair is a critically important area of research at the moment. The C-Trap is ideally suited for understanding these mechanisms of action. In the paper shown here from the laboratories of Simon Bolton, Simon Powell, and David Rueda, they studied a DNA helicase called HELQ. Defects in this helicase are implicated in various cancers. The researchers used the C-Trap to grab individual DNA molecules and visualize individual proteins on the DNA, as shown in the highlighted image. By moving this complex using the microfluidics and exposing them to different transcriptional activity and cofactors, they were able to unlock a new understanding of this fundamental biological process. Another critically important area of research is understanding the processes related to production and folding of proteins in the cell and how they can go awry in disease. In the Nature Communications publication by Sandra Tanz and Alexander Katranaitis, these researchers leveraged the incredible biophysical sensitivity and correlated fluorescence imaging of the C-Trap to better understand how these proteins fold while they're produced by the ribosomes. By pulling on the C-terminal and N-terminal domains of a zinc finger protein using the optical tweezers, the researchers were able to repeatedly and precisely measure the forces folding and unfolding within the ribosome, while simultaneously measuring the fluorescence. Overall, these data from the same experiment provided two independent and direct observations of protein unfolding and correctly refolding within the ribosomal tunnel. The last highlight here from the Banerjee lab in a protein science article illustrates how the C-Trap's multi-channel fluorescence combined with optical tweezers can be used to identify an important mechanism of action in biomolecular condensates. In this figure, They were able to form a condensate on a DNA molecule and then show the recruitment of a transcriptional activator into the molecular condensate using these dual fluorescence channels. You can see here in the image the co-localization of the two species. This was a powerful example of ability to directly observe the mechanistic interactions of the C-Trap. So by providing a fully integrated and, more importantly, very easy to use system, our customers are usually up and running in under a week. This allows them to focus solely on generating science. Most of our customers are able to publish their first impactful science in well under 2 years, and then on average, every 10 months thereafter. So we successfully brought our C-Trap technology to the most innovative biophysics and biology labs over the past several years. This is based on correlated combination of tweezers, confocal and STED microscopy, and microfluidics. This has been an especially powerful tool for complex assays in solution where the molecules have been manipulated, measured, and visualized, like in DNA binding proteins, protein folding experiments, and phase separation application. However, today we are really excited to introduce a new member to the C-Trap family. Are you ready? That was really nice. So for the last several years, there's been a really important need for a tweezer system optimized for assays requiring imaging at the surface. In many areas of research, such as cytoskeletal structure, intracellular transport, mechanobiology, they require a unique set of capabilities to dynamically interact with but measure at the surface of the flow cell. After several years of development and support by many key collaborators, we're really excited to announce today the new C-Trap Edge platform. This is a C-Trap wholly suited for the surface assays with powerful optical tweezers, microfluidics technologies, combined with a unique set of capabilities of imaging at the surface. With this announcement, we're also rebranding our flagship C-Trap for solution assays to the name C-Trap Dymo. With both, we're absolutely committed to the ongoing development of these product lines with hardware, software, and biochemical reagent innovations so that you can focus and get the most out of your research. We are really excited to be launching this new tool today. I'm now going to hand this talk over to Dr. Trey Simpson, head of our application scientist team. He's going to dive a little bit deeper into the incredible new capabilities of the C-Trap Edge and how they address key technical issues with studies at the surface. Remember, following Trey will be Dr. Arne Gennerich to share some of his science of microtubules and motor proteins. Thanks very much.
Thanks, Trevor. So as Trevor said, I'm going to take you through a deeper dive into the C-Trap Edge and what makes it optimized for surface-based biological assays. So to start, let's take a look at a cell. As you know, at all times, there are many complex biological processes going on within the cells. There are organelles sending signals to other organelles, proteins are being made. There's this interweaving complex network of cytoskeletal filaments, along which motor proteins can carry cargo from one end of the cell to the other. If even one of these processes goes wrong, it can lead to catastrophic diseases and potentially cell or even organism death. And to truly understand how these diseases work, we have to look at the most fundamental level. Historically, the tools to do this have been complex and really reserved for the most expert labs that could build their own systems. And today I'm going to talk a bit about how the C-Trap Edge will allow anyone to do these experiments. To do so, we're going to look at a motor protein called kinesin that walks along microtubules. To give you a sense of what this looks like, here's a video of a kinesin walking along a microtubule. As you can see, it really does look like it walks. It seems to take these individual steps one after another, picking up one foot and putting it forward and stepping back on the microtubule again. In this case, it's also carrying along a cargo. There's a lot of things here to understand, especially when we zoom into the single molecule level. To do that, we're going to form a single molecule assay on the surface. Here, we have a microtubule on the surface and a cargo that this kinesin will pull along. There's a lot of things we're interested in here to really understand how this process works. We want to be able to see step sizes and substeps of this kinesin motor, what you saw in that video earlier. We want to know how the mechanical load of this cargo affects the way this motor protein works. We want to know how it processes along, how fast it moves, what type of steps it takes, and even the forces that these individual steps generate one after another. To do this, we can use the C-Trap edge, and particularly optical tweezers. But what you may notice if you're familiar with optical tweezers is as you get down near the surface and try and do these complex single molecule assays on the surface of your chamber, you run into a lot of challenges. Particularly, you need really accurate trap positioning control to find these microtubules and bring a motor protein in contact. You still need to measure an accurate force, which gets much more challenging near the surface due to difficult calibration procedures. You also need a stable surface to be able to measure over longer periods of time, necessary to really understand how these motor proteins move from one end of a cell to another, or really from one end of a filament to another. We've solved many of these issues for these surface-based assays with the C-Trap edge. We're able to visualize cytoskeletal filaments and motor dynamics using turf imaging and IRM. We can actually see individual substeps of cytoskeletal motors due to our accurate force calibration method near the surface. We're even able to see force generation and the effect of load on the way this motor protein functions. All of this is combined with dedicated workflows into a correlated software package to give you the fastest time to impactful results. So, let's dive a little deeper into what this looks like on the C-Trap edge. On the left here, you'll see a turf image of microtubules and also kinesin motors. In this case, the microtubules are labeled in red and the kinesin motors in blue. And you can see them walking along the length of these filaments. What you may also notice in this case is that this red dye on our microtubules photobleaches over time. And in some cases, you may actually have a filament, microtubule, or whatever it may be, that you can't label at all because maybe the labeling interferes with the biological processes you're interested in. For those cases, we actually have IRM imaging. IRM allows you, at the surface of your chamber, to see individual filaments and any height changes in these filaments without labeling at all. In this case, we're able to see these microtubules that are as small as 25 nanometers in diameter. But this is also correlated with the turf imaging. What this allows us to do is locate, see these microtubules on the surface, but also see kinesin motors visually walking along these filaments. In this case, the kinesin motor is still labeled in blue. You can see walking along the filaments here. What this allows us to do is we can look at many of these filaments and many motors at once and see how they move along, how they process, what speed they move, whether they go forward or backwards, all the information we're interested in visually in one assay. What we can actually do is look along a single filament here and track the motor proteins, the kinesin, walking along. And this is as simple as a click of a button. We draw a line along the microtubule, pull the imaging data out, and then click on our tracking software. This finds each of these individual motor proteins and shows you as to how they've moved along. As you may see, there's some cases where they step forward, some where they reverse, and some where they go on these runs where they go really quickly along the filament. We can do this over a field of view, one filament at a time, and track many motor proteins. This gives us good statistics to be able to determine the velocity of these motors. In this case, these kinesin are moving at about 300 nanometers a second. But, as we mentioned earlier, we're also interested in the forces that these motor proteins are generating and seeing the details of even the substeps of these proteins. In this visual case, we can see the individual steps in a direction, but there may be some hidden detail that we're missing. So, this is where we bring back in the optical tweezers we mentioned earlier. But as you may remember, there were some challenges. We need to be able to have an accurate force calibration near the surface to be able to make the proper conclusions on the forces this motor protein generates, and to be able to accurately see the individual steps it takes. We have solved the calibration issue in the C-Trap edge. In this case, Historically, when you would calibrate optical tweezers, you would look at the thermal fluctuations of the bead within the trap. But you also have to manually input some parameters. You have to input things like bead radius, viscosity, temperature. All of these are components of drag. Manually entering them is making some assumptions, that the radius that you're told by the manufacturer is correct, or that you really know the viscosity of your solution. But also on top of that, as you get closer to the surface, the relative drag coefficient on the particle you have trapped changes drastically by up to 60%. This makes your calibration highly dependent on the distance you are from the surface. As a result, when you go through this calibration process and get a stiffness of your optical trap, this change in drag coefficient near the surface has a drastic effect on the stiffness you measure as well. We would expect a constant stiffness as we get closer to the surface, but our calibration shows us that this stiffness seems much smaller at the surface than it is in reality. To solve this calibration issue, we introduce an oscillation. This oscillation allows us to directly measure the components of drag as opposed to assuming them. When we directly measure these components of drag and then use these in our calibration, the stiffness that we measure is completely independent of height to the surface. As a result, we get an accurate trap stiffness and an accurate force measurement at any height within our chamber. This will enable us to take accurate force measurements of our kinesin motor walking along a microtubule that's sitting along the surface here. So, how can we use this? So let's talk about a couple experiments we can do with these kinesin motors and microtubules. In this case, we're going to trap our particle, or our cargo, that our kinesin motor is attached to, put it in contact with our microtubule with some simple workflows as simple as clicking on the microtubule on the screen. And as this motor protein tries to walk along, it's going to pull this cargo out of the trap. This will show up as a force in our force readout. And what you see on the left here in this curve is the force begins to increase as this motor protein starts to walk. But at some point it stops. This is typically known as the stall force, and in this case is around 5 piconewtons for this motor. Once it's reached the stall force, we'll start to see these individual steps back and forth. And what we can actually look at here is we can zoom in to this section, looking at these steps, and we can see the individual steps where this motor goes forward and backwards, reverses, stalls for a while, and also see the forces that's generated each time, in this case as small as half a piconewton. This gives us a really good sense of these individual steps of the motor with the forces they generate, but in this case we don't really see the processivity of the motor. For that, we can use a different assay. Here, we're still using the optical tweezers and the C-Trap edge, but we're going to bring our cargo and motor protein in contact with the microtubule, and instead of letting it walk out of the trap, we're going to follow it along. In this case, this is typically referred to as a force clamp, an easy to use feature on the C-Trap where you just set the force you want to clamp at, click the button, and what will happen is our trap will follow along as this motor protein generates force to keep a constant load on the cargo and the motor protein itself. This allows us to determine the effect of the load of this cargo on the way this motor protein functions, but also allows us to track the processivity of this motor over the entire length of a filament. This is also where the surface stability I mentioned earlier is critical. As we move along this filament, we can't have the surface drifting, and we need to know that the entire movement we're seeing here is due to this motor protein walking. What you'll see here is over time, our bead position changes, and this directly correlates to the walking of our motor. We see these individual steps, we can look at all of them, put them together, and actually see that our individual step size is 8 nanometers. And we are able to do this over the entire length of a filament. Now, in this case, we're mostly talking about forces in the X and Y direction. This kinesin motor, in the assay we set up, mostly generates forces along the length of our microtubule or in the direction parallel to the surface of our chamber. But in some assays with different geometries or maybe different motors you're interested in, there may be forces generated in the Z direction or the direction perpendicular to the surface. For this, the C-Trap edge introduces axial force detection. This allows us, in just as accurate as our measurements we've shown here and with the same resolution, to measure forces in the direction perpendicular to the surface. This can open up a whole new world of assays, of biological assays, using different configurations for your motor proteins here and the filaments, as well as making sure you fully understand the way this motor protein generates its force. So, what does this actually look like on a C-Trap? I'm going to walk you through a bit here. One of our scientists that you've seen earlier, putting this sample in a C-Trap and running through an experiment we've discussed earlier. So to start, he loads his sample into a chamber and places it on the C-Trap edge. He slides it on and it snaps in the back, and he's ready to go. Does a nice little spin and gets to work. So now that he's on the system, has his sample loaded, all the controls are as simple as a couple mouse clicks and some tilts of the joystick. In this case, we're going to do the force generation assay we talked about earlier, where he's going to move, using the joystick in this case, his cargo in contact with the microtubule, and then let this kinesin start walking along and applying force to our cargo. What you'll see on the left here is a similar curve we saw earlier. Force is generated, increases, and then stalls for a while and takes some steps forward and backwards. In this case, you also see this sort of immediate jump back to a lower force. What's happening here is our motor proteins, contacts of the microtubule, are actually getting pulled apart. At some point, the force is too great for this motor protein to stay attached to the microtubule. It falls loose, and our cargo snaps back to the center of the trap. But immediately after that, the motor protein starts working again, starts walking, and we see an increase in force. This can be repeated over and over again, and data continue to be collected. Okay, so we've taken a deeper dive in the C-Trap edge and how it's optimized for surface-based biological assays. We've discussed how you can visualize these filaments with turf and see motor proteins walking along, but you can even see them label-free with IRM. We've solved the calibration challenges near the surface of the chamber to where you can now have an accurate force measurement all the way down to the surface, and allows you to see these individual steps of cytoskeletal motors, the forces they generate, the effects of load on their way they work, and truly understand how these processes work at their most fundamental level. As you saw, all this is combined into a nice, user-friendly, easy-to-use software, but also with dedicated workflows that allow you to have the fastest time to the most impactful results. Overall, as Trevor mentioned earlier, our goal is to democratize these tools for all researchers in the field. We hope to truly unlock single molecule analysis for biologists, physicists, chemists, really anyone around the world. We truly believe that this will lead to fundamental discoveries that allow us to better understand disease, hopefully develop better cures, and overall lead to the betterment of human health. I've mentioned, how this can work with some surface assays, particularly involving filaments and motor proteins, but there are many other uses here. For example, if you're interested in cell membrane mechanics, you could have a cell on the surface, pull on the membrane, see how it responds, and now you can do that in a very accurate manner due to the new calibration. But I haven't even mentioned that the TURF and IRM can be combined with wide field imaging as well. This wide field imaging allows you to see down to single molecules. This could be useful for studying DNA binding proteins and their role in cancer, or protein folding and protein dynamics, and seeing how proteins that misfold could be relevant in disease. It also allows you to study things like phase separation and see how two droplets fuse, the properties of each droplet and how the rate and how they fuse, which also is a big hot field of study for many diseases today. Overall, we hope the C-Trap Edge is a versatile tool for your lab and other researchers around the world. So now, I'm excited to hand it over to Professor Gennerich, who's going to talk to you about how he has used the C-Trap Edge and some surface-based assays to investigate motor proteins that his lab is interested in.
Hello, everyone. I'm going to tell you about our recent structure, function, and mutagenesis studies we performed to determine the motility and force generation mechanism of the kinesin motor KIF1A. As you know, or as most of you know, kinesin motors step along microtubule filaments. They almost walk like humans in a step-like fashion, hand over hand, along a microtubule protofilament by hydrolyzing one ATP molecule per step. In contrast to the founding family member kinesin 1, KIF1A belongs to the kinesin 3 family of microtubule motors. To the same family, KIF 13, 14, 16, and 28 belong. Also, like kinesin 1, KIF1A is a plus interacted motor. It moves in the antegrade direction of microtubules in neurons because it's expressed in neurons. But in contrast to kinesin 1, KIF1A has one particular function which is quite intriguing. It moves the nucleus of dividing stem cells in the brain, the so-called radioglia progenitor cells, which give rise to numerous different neurons in the brain. But KIF1A is also involved in the transport of cargos like vesicles towards the synapse in neurons. Because of its essential functions during brain development, it is not surprising that mutations in KIF1A cause disease, in particular neurodevelopmental diseases and neurodegenerative diseases, which have been recently coined KIF1-associated neurological disorders, for short KAND. And because there are no cures available, and these kids often die after half a year or year because of the progression of the disease and degeneration, parents of those children have founded KIF1A.org, a non-profit organization that provides the framework for families worldwide to come together to discuss the problems of their children, but also to facilitate and promote the development of therapeutics. KIF1A is composed of about 1,700 amino acids. You can see here on the left is the motor domain. In violet, there's the NL is the neck linker, which generates force, and neck coid, which is essential and sufficient for dimerization of the motor, but also a pH domain for binding to lipids. I show you here data which are not up to date, but they show that most patients who have mutations in the motor domain, while mutations can occur along the entire gene actually. So I show you here a few mutations which are in the motor domain, and some of them you can immediately see in helices that are interacting with the microfilaments. So you can imagine that the interaction of the motor with microtubules is impaired or affected by these mutations, and therefore might even have effects on force generation and motility. The questions we want to address are: how much force does KIF1A generate? Why is KIF1A super processive? And what I mean with it, I will show you in a minute. Why is KIF1A so fast? It moves with on average 2 micrometer per second, which is more than twice as fast as kinesin 1, for instance. And also KIF1A has a unique structure, an element in the motor domain, the so-called K-loop, because it's rich of a high number of positive charged lysines that interact with microtubules. And obviously, because my lab is at the medical school, we want to do some translational research. We determine the effects of disease mutations, and we also work together with companies that try to develop therapeutics for KAND. We use for our work the LUMICKS C-Trap. Here I took a picture which makes it look nice. It's in our basement. This instrument allows us to do correlative measurements of motility and force generation. We have a 3-color turf microscope combined with 2 optical traps, but also, as you can see here, a microfluidic cell with up to 5 channels in our case, where we can even do drug screening, which we do currently, to see whether drugs can reactivate a partial wild-type function in these mutants. For our work, initially we started out to use a tail-truncated motor, which is easier to express, in this case, even in E. coli bacteria. Importantly, it has the neck coil, which is sufficient for dimerization. But because in our measurements where we work typically at picomolar concentrations, sometimes the motors can fall apart, form monomers. So we have placed also leucine zipper, which really keeps the motor in a dimerized state. So when we use this motor and fluorescent label it, we can use a turf microscope and visualize the movement of these motors along microtubules. As you might know, you can draw a line along the microtubule that cannot be seen here and stack it over time. If you do so, you can create a kymograph. And you can see here the long lines, the diagonal lines, which show how processive KIF1A molecules are. You see the scale bar down here of 5 micrometer. These motors move over more than 20 micrometer distance, which is really, we call it super processive, which was coined actually by Kristen Wehe's lab in 2014. So in addition, the C-Trap, we use it in the single beam mode or a single beat mode. So you all know about trapping. What we did, we started out with force measurements on kinesin 1 to set the stage, to really show that the C-Trap works and generates forces as expected. Kinesin 1, in this case, KIF5C, generates 5 piconewton, and this is the same force that we measure also with the home-built optical trap. And this is also a take-home message, because often if you look at literature, it is reported that kinesin 1 generates 6 or 7 piconewton, but we can say that the true force generation of kinesin 1 is about 5 piconewton. In contrast, when we did the first force measurements on KIF1A with the C-Trap, we were really surprised. We saw these trains of force generation events, the clustering of force generation events, which almost looks like a sawtooth pattern. And we were wondering, what is going on? So we thought it is really important to exclude first that we work with aggregated proteins, which might increase the on rate, as seen here, or might underlie the rapid rebinding. And we needed to exclude that we work at a multimotor, at multiple motor level to find the appropriate concentration. So to rule out aggregation, we used the fluorescent capabilities to look at photobleaching of these motors, and we could show that the motors mostly bleach in 1 or 2 steps, which suggests that we are dealing with dimers and not in higher order oligomers. But importantly, because there were no force generation reports on KIF1A at least, we needed to make sure and provide evidence that a single motor is capable of generating the forces that we see, which means we measured the fraction of moving force-generating beats as a function of the relative motor concentration. So then we can fit 2 models, one that is derived for 1 or more motors, or another model with 2 or more motors, so that we can provide evidence that 1 motor is sufficient. And you can see here, the solid line describes the data better, which is for 1 or more motors. In addition, and as support that a single motor X generates the forces, is that we observe only 8 nanometer steps and not degraded steps, which could be expected for multimotors driving beat movement. So this put us in some corner, because a published science paper published in 2002 from Ron Bale has shown that the ortholog of KIF1A, which is Ankanat 4 from C. elegans, supposedly generates forces of up to 6 piconewton, like kinesin 1. So we were wondering, what is going on? Is it true that Ankanat 4 generates more force than its ortholog KIF1A? Because sequence-wise, they're really quite identical. Not identical 100%, but really quite well identical. So when we recombinantly express this motor, Ankanat 4, 389 amino acids, again with the leucine zipper, forced or stabilized dimerization, you can see that we also measure 3 piconewton. Around 3 piconewton, which is a force we also saw for KIF1. So if we look back at KIF1A, similar construct, we see sometimes stalling, but it doesn't stall often. So we were thinking, how can we determine what the maximum force generation is, or what the stall force is? And to introduce an arbitrary time duration for which a motor needs to stall to count its stalling, you know, it's arbitrary. So what we did is we analyzed the forces at which the motor stalls at a given time frame or time duration as a function of the criterion. for how long a motor should stall. And you can see that the stall force does not depend on the definition, what we call stalling. And this really nicely shows that the maximum force generation of KIF1A as Ankanat 4 is indeed 3 piconewton, significantly less than kinesin 1. But there's one notion. The lab of Mike Ostap at UPenn published a beautiful biophysical journal paper 2 years ago, in which they showed that the force generation of kinesin 1 depends on the geometry, what kind of assay one uses. And as we use a single bead assay, we were wondering whether we observe the forces because we might use the wrong assay. So Mike Ostap's lab has shown that if you turn the assay around, where you have a polystyrene bead on the cover glass and then lower the microtubule with the 2 traps, where the angle between the kinesin motor and microtubule can be even 0. While in our assay, in the single bead assay, the angle between the motor and the microtubule is determined by the bead surface, which suggests, in their case, that the likelihood for a motor to stall strongly depends on the z-force, the axial force. And you can see here, for instance, that kinesin 1 barely stalls, detaches before it reaches maximum force generation in the single bead assay. But in the 3 bead assay, where the angles are really small and the z-force is really small, they almost see stalling all the time. So if we look back at our data, we realize we work with a tail-truncated motor. So a short truncated construct, which is significantly shorter than the full-length motor. So we were wondering whether a longer motor, which has a smaller z-force component than a short motor, we were thinking, okay, we also have to do the measurements with the full-length motor. So for the full-length motor, we used a motor expressed in mammalian cells. And as you know, or as you might know, full-length wild-type KIF1A is out-inhibited, usually. And we hoped, or we hypothesized, that bead attachment might relieve the out-inhibition and you might observe force generation, but it wasn't the case. So we bound these motors to polystyrene beads, used it in the C-Trap, but we could barely see any force generation, just short, brief interactions, forces up to 0.3 piconewton, but also in both directions. So in order to really determine what force full-length KIF1A generates, we used a trick. There is a mutation, the V483 mutation, in the first coil domain of KIF1A, which has been shown also by Kristen Wehr's lab, that this coil domain is involved in outer inhibition and that this mutation relieves the outer inhibition. So we use this motor, and to our not surprise, but we were really happy to see that this motor is fully active, has a high on rate as the tail-truncated motor, and moves up to 3 piconewton. And we also, in this case, did an analysis as a function of the stalling criterion, the time frame, and you can see that also the wild-type full-length motor generates 3 piconewton. And this means that the angle, at least the small angle change, because of the different lengths of the wild-type of the full-length motor compared to the tail-truncated motor, does not affect the total force output of the motor. However, when we measured the stalling time, which is really brief for KIF1A, in contrast to kinesin 1, we see that the wild-type full-length motor, that the full-length motor stalls indeed for a significantly longer time than the tail-truncated motor. And this suggests that the z-force indeed affects the stalling time of KIF1A. So we answered now how much force KIF1A generates, 3 piconewton. But now the last question I want to ask, what is KIF1A's class-specific K-loop doing? What is its function? How is it involved in force generation? So the K-loop, as shown here, it's called K-loop because there are 6 lysines in loop 12. And the K-loop, or loop 12, is interacting with the microtubules. So it's positively charged and is interacting with the negatively charged carboxyl terminus of tubulin. And so and it has been shown that the K-loop is important for the initial binding of KIF1A to microtubules in the ADP state. So we are wondering whether the rapid rebinding of KIF1A during fourth generation is also dependent on the K-loop. So what we did is, if you look at kinesin 1, KIF5C here is again kinesin 1, you can see that kinesin 1 has only one lysine compared to KIF1A. So we created two swap mutant where we replaced the loop 12 of KIF1A with the loop 12 of kinesin 1 or KIF5C. And then in addition, we removed the positive charge that is left there and mutated in alanine, which is not charged, to see how the binding changes to microtubules during fourth generation. And what you can see here is remarkably indeed the rapid rebinding. You can see there are only single events and not multiple bindings directly after another, show that the K-loop is responsible for the rebinding. And more quantitatively, you can see here for kinesin 1, which is shown down here, the rebinding rate or the number of events per microtubule encounter, so which we call the clustering, and the number that defines the clustering of the fourth generation events. Kinesin 1 only shows on average 1 point something events per encounter. So you see mostly single events. The wild type motor, whether it's a tail truncated or full length motor, show 30 events per microtubule encounter, which is significantly higher. But with the swapping of loop 12, you can see that the rates of rebinding, the number of events, dramatically going down. Because this is a logarithmic scale axis, you can see this is maybe even something like 2 events or 2.5 events or so per encounter. So this means the K-loop is involved. And a last thing I want to show you is, because I mentioned Kant, the disease, which is called the mutations in KIF1A, there's one mutation here, the proline 305R mutation, which causes Kant, which is adjacent to the K-loop. And we wanted to know what happens actually if you induce this missense mutation. And you can see here in the data that indeed the P305R mutation dramatically affects the force output of the motor. It also affects the velocity, 50% down, as well as the run length of the motor, which is also roughly 50% down. I also list here the data of a different mutant, the R316W mutation. And again, this is a mutation which is close, again, to the K-loop. So they phenocopy so, but you can see that this mutation even has a more dramatic effect. Okay, just to summarize what we have learned, kinesin 1 generates about 5 piconewton. KIF1A and Ankanat4 generate only 3 piconewton. In contrast to other motors, such as the kinesin motor OSM3, which we showed long time ago, is activated by beat attachment. KIF1A autoinhibition cannot be relieved by cargo attachment alone, or by beat attachment in this case. But if we induce mutation in the first colloid region, which relieves autoinhibition, we see beautiful fourth generation and the sawtooth pattern of events, which is really unique for KIF1A. Yeah, and I also showed you that the K-loop is the main driver, or is mainly responsible for the rapid rebinding during fourth generation. And as kinesin 1, KIF1A also generates 8 nanometer center of mass steps. Lastly, I would like to thank in particular my research assistant, Professor Lura, who did all the force measurements with the C-Trap and the mutagenesis studies, structure function studies, and my collaborators on this particular work, Christian Wehe and David Zepp from University of Michigan. And I also would like to mention my other friends and collaborators on KIF1A, Hernando Sosa. We do some structural work on KIF1A. Rick McKinney at UC Davis. We also published recently a paper together where we also use the C-Trap for our work. And Wendy Shang, who is the main clinician in the world for kids who have KIF1A, and Richard Valley and NIH funding, and in particular KIF1A.org, who are also supporting this work. And LUMICKS, obviously, in particular for the assistant for all problems one can have with an assay and so forth. Thank you very much for listening.